Good morning, everyone. Am I audible?
Yes,
Yes,
sir.
sir.
Good
Yes,
morning,
sir. Good
sir.
morning, sir.
Good morning, sir.
Okay, everyone can see the slides.
Yes, sir.
So we will start today's class. So in last class, we discussed about fatty acid biosynthesis. Today we are going to study about fatty acid oxidation. So biosynthesis was basically how fatty acid is formed, and this is opposite, but it's not directly opposite. Here we will see how fatty acid can be broken down to release energy. Okay, normally glucose is the primary source of energy, but if the glucose storage depletes, gluconeogenesis cannot happen or it has exhausted, then fat starts breaking down. So then, especially in starvation and all, fatty acid oxidation is very important to provide energy. So this we have already discussed. Fatty acid is basically a long hydrocarbon chain and a terminal carboxyl group, right? This is the long hydrocarbon chain with carboxyl group at one end. And the chain can be saturated, unsaturated, these things you have discussed all. Okay. And from where these fatty acids comes. So this we discussed, either you can take directly in diet, okay, or adipolysis by basically by breaking down of triacylglycerol. Triacylglycerol contains glycerol and three fatty acids. So by the breaking down of triacylglycerol, these fatty acids can be formed either in the tissues like liver and all or in the circulation, or it can be the de novo synthesis like this fatty acid, as we discussed in last class. It can be synthesized. De novo means new synthesis can happen. Okay, so first the overview of the fatty acid oxidation. It is opposite, but it is not a simple reversal of biosynthesis. So don't think that those reactions which are involved in biosynthesis, if you do those reactions in opposite direction, fatty acid oxidation can happen. That does not happen in body. Now fatty acid biosynthesis happens in cytosols, as we discussed, but this oxidation of fatty acid takes place in mitochondria. So this is very important that it is compartmentalized basically. There we saw that in fatty acid biosynthesis, the main enzyme was fatty acid synthase, which was a complex of 7 enzymes, which was encoded by one single gene, okay, one single polypeptide with functional division. But here all the enzymes involved in the fatty acid oxidation are individual. So individual regulation will happen with integration with tissue requirement. Depending on the requirement in particular tissue, this fatty acid oxidation will happen in that tissue. And this fatty acid oxidation is an aerobic process. It requires presence of oxygen. Okay, so this is important. And what are basically the cofactor or the intermediate or the substrate required for this are acyl coenzyme derivative. We'll see what is acyl coenzyme. Acyl coenzyme is nothing but fatty acid which is combined to coenzyme A. So that is known as acyl coenzyme A. So first fatty acid has to combine with coenzyme A, then only the oxidation of fatty acid can happen. So that acyl coenzyme A will start forming different derivatives, and which utilizes two cofactors, NAD plus and FAD, and ultimately it will generate ATP. That is the whole purpose of oxidation of fatty acid. Okay. Now there are many pathways by which fatty acid oxidation can happen. We saw that biosynthesis happen by one pathway, right? But this oxidation can happen by many pathways. The major pathway, the most important pathway is known as beta oxidation. Others are alpha oxidation, omega oxidation, and paroxysmal oxidation. So beta oxidation is the major mechanism which occurs in the mitochondrial matrix. So you remember the site. Okay. And here two carbon units are released as acetyl coenzyme per cycle. There we saw in biosynthesis acetyl coenzyme was the main substrate required, right? That was first formed in alanine coenzyme, and ultimately further reaction of fatty acid biosynthesis was happening. Here in oxidation, acetyl coenzyme A will be released after one set of cycle, and depending on the chain length of the fatty acid, it will require many cycles for all the acetyl coenzyme A to be released. Basically, to be for the breaking down of fatty acid, depending on the chain length, the cycle will repeat. Okay. Alpha oxidation, it mainly takes place in brain and liver. Okay. Here one carbon is lost in the form of CO2 per cycle. Omega oxidation, it is a minor mechanism, but if in some if in someone beta oxidation is impaired, we will see the condition also when the beta oxidation gets impaired in clinical significance we will read, and in that time this omega oxidation becomes important in the body. Paroxysmal oxidation, this is mainly for if the chain length of the fatty acid is very long. To decrease the chain length, this oxidation, this type of oxidation happen in paroxysm. Okay. Mainly today we are going to discuss the details about beta oxidation. Okay. Others are only minor pathways in the body. So the once the fatty acid, I told you that it has to first combine with coenzyme, right? That is known as activation. Once the fatty acid is activated, it means that acyl coenzyme has formed. The further reaction will go into the sequence of 4 reactions. Okay. So saturated acyl coenzyme is dictated by a recurring sequence of 4 reactions. Recurring sequence. So this sequence of reactions will not happen one time only. Depending on the chain length, this sequence of reaction will repeat many times. So first activation into acyl coenzyme, then oxidation where FAD will be required. So remember this thing, very simple. Then hydration, then again oxidation, but this time the cofactor required is NAD. Okay. Then thiolysis will happen with the help of coenzyme SH. Right. So first is activation into acyl coenzyme. Okay, I'll repeat many times. You can remember now only. Then second is oxidation with the help of FAD. Then hydration. Hydration means water molecule will be added. Then oxidation by NAD plus. Then thiolysis. So basically total 5 steps are happening, but suppose a fatty acid has 16 carbon chain, right? So there the activation will happen once only. It will form 16 carbon acyl coenzyme A chain of the fatty acid. Then oxidation will happen, hydration will happen, oxidation will happen, thiolysis will happen, ultimately leading to the release of one acetyl coenzyme, one 2 carbon unit, which is acetyl coenzyme. Then the 14 will remain, right? One acetyl coenzyme has released, then again this whole 4 reaction will again happen, again 2 more will go, then 12 carbon will be left. So like that, the cycle will repeat as long as there are more than 2, basically there are more than 2 acetyl coenzyme, 2 carbon in the chain. We'll see the reactions. Fatty acyl chain is shortened by 2 carbon atoms as a result of these reactions. So these 4 reactions will lead to release of acetyl coenzyme, leading to the shortening of the fatty acyl chain by 2 carbon. Okay. So FAD and NAD is used. What will it form? FADH2 will form, NADH will form, and acetyl coenzyme will form at the end of thiolysis. Why it is called beta oxidation? The reason is that oxidation is on the beta carbon. What I had told that from the COOH side of the fatty acid, after COH, the first carbon is known as alpha, second carbon is known as beta. So the chain is broken between alpha and beta. Because it is between alpha and beta, that's why this reaction is known as beta oxidation. Alpha oxidation will be basically the chain will break from the COH and alpha carbon, between COOH and alpha carbon. Then that will be alpha oxidation. Okay. Okay, so how the activation of fatty acid happens? This is the first reaction, right? Then only the proper oxidation of fatty acid can happen. So activation of fatty acid. Basically, it is two-step process, and this is the only step in the whole reaction where ATP will be required. So ultimate goal of the fatty acid is to release energy, right? Release ATP for the energy purpose in the body. But in the activation part, it still needs ATP. So how this fatty acid is combining with ATP and forming acyl adenylate. Okay, plus 2 high energy phosphate bonds are released, right? So what is happening? How many ATP is used here? It seems like 1 ATP is used, but it is equal it is ultimately forming AMP releasing 2 phosphate, right? This high energy phosphate. So normally from ATP to ADP is formed. So we say that 1 ATP was required. Here equivalent of 2 ATP is required because it is forming AMP, 2 phosphate are required. So that's why if they ask in exam or when you write the whole reaction, how many ATP are required? It is equivalent to 2 ATP. It is needing 1 ATP but it is forming AMP. So for it to form back to ATP in the body, 2 phosphate will be required, right? 2 high energy phosphates ultimately, which is equivalent to 1 ATP. So fatty acid must first be converted into active intermediate before they can be catabolized. This is the only step in the complete degradation of fatty acid which requires energy from ATP, as I told. So fatty acid plus ATP forming acyl adenylate, and this acyl adenylate combines with coenzyme ASH forming acyl coenzyme A. So acyl coenzyme A is nothing but fatty acid combined with coenzyme ASH, and then AMP will be released. So now acyl coenzyme A has formed, and then only fatty acid oxidation can happen. So now this fatty acid are activated on the, this is important, where fatty acid is formed, fatty acid is forming cytosol. Where fatty acid is activated, it is activated on the outer mitochondrial membrane. Okay, but the oxidation where it will happen in the mitochondrial matrix. So then, as the fatty acid has got activated in the outer mitochondrial membrane, it has to somehow get transported inside the mitochondrial matrix. So how it gets transported? First, you have to read that. First, activation of fatty acid happening at the outer mitochondrial membrane. Then the transport of this activated fatty acid into the mitochondrial matrix, and then only the oxidation of fatty acid can happen inside the mitochondrial matrix. So for the transport of activated fatty acid from the outer mitochondrial membrane into the mitochondrial matrix, carnitine is required. Okay. So activated long chain fatty acid are transported across the membrane by conjugating them to carnitine. So basically activated fatty acid combines with carnitine at the outer mitochondrial membrane and it goes inside the mitochondrial matrix. Carnitine is deuter ionic alcohol. So here acyl coenzyme A, okay, so this is basically what? Activated fatty acid, right, at the outer mitochondrial membrane. Its coenzyme A group combined with the hydroxyl group of the carnitine. Okay, so carnitine is hydroxy trimethyl ammonium butyrate. If you don't know, it's fine. It is widely distributed and particularly abundant in muscle. Carnitine is obtained from food, okay, mainly animal-based food, or it can be synthesized in body also. So how it is carrying, next we will see that how this carnitine helps in carrying the activated fatty acid, which is known as acyl coenzyme A, from the outer mitochondrial membrane inside the mitochondrial matrix. You read this slide also carefully. The acyl group combines with the hydroxyl group of carnitine to form acyl carnitine. Okay, so this acyl is combining with carnitine forming acyl carnitine. This reaction is catalyzed by carnitine acyl transferase 1. So now depending on the fatty acid combined with coenzyme A, if the fatty acid which has combined is palmitic acid, then it will be acyl palmitoyl carnitine palmitoyl acyl transferase. The name will depend on that. So CPT1, because we normally read, because we cannot read like metabolism, either biosynthesis or oxidation for each and every fatty acid present in the body, right? So we take as a model, as an example, palmitic acid, which is very much abundant. So that enzyme will be named as carnitine palmitoyl acyl transferase 1, CPT1. Okay, so this enzyme helps in the combining of palmitoyl acyl coenzyme A with carnitine, forming palmitoyl acyl carnitine. Next, this acyl carnitine, where this reaction will happen? Outer mitochondrial membrane, right? Because there the activated fatty acid is present, acyl coenzyme A. So this acyl carnitine is then shuttled across the inner mitochondrial membrane by a translocase. So from outer to inner mitochondrial membrane, another enzyme will help in this transport basically. So that is, this translocase is acting as transporter. Okay, so this translocase will transport this acyl carnitine from the outer mitochondrial membrane to inner mitochondrial membrane. Now in the inner mitochondrial membrane on the matrix side, this acyl group is transferred back to coenzyme A. From the carnitine, this acyl group will again go back to coenzyme A, forming acyl coenzyme A in the matrix site of the inner mitochondrial membrane, and this reaction will be catalyzed by carnitine acyl transferase to CPT2 in case of palmitic acid. Finally, the translocase returns carnitine now to the cytosolic site in exchange for an incoming acyl carnitine. So this cycle will keep repeating. See here, this is fatty acid, this is plasma membrane. Okay, so this is cytosol. Just think. Okay, this is a symmetric diagram only, right? So this fatty acid can either, I told you the source. What are the sources of fatty acid? Either you can take in diet. From that diet, it will go inside the cell. Okay, right? After breaking out of triacylglycerol, or it can be synthesized new, right? So it can fatty acid, we saw that fatty acid biosynthesis can happen in the cytosol, so it can be synthesized new also. So those fatty acid on the outer mitochondrial membrane, after getting activated, it will be forming what? Acyl coenzyme A, right? This activated fatty acid. Then acyl coenzyme A combines with carnitine. So acyl coenzyme A combines with carnitine, forming acyl carnitine with the help of CPT1. Okay, carnitine palmitoyl acyl transferase 1. This acyl carnitine, this is translocase. Okay, will be transported across the inner mitochondrial membrane on the matrix side of the inner mitochondrial membrane. It will go, then this acyl carnitine again will be converted back to carnitine plus acyl coenzyme, right? So this acyl coenzyme will be utilized for beta oxidation in the matrix, mitochondrial matrix, and this carnitine will again transported back to the outer mitochondrial membrane with the help of translocase enzyme. In this whole thing, what all you have to know, fatty acid is present in the cytosol. At the outer mitochondrial membrane, it gets activated into acyl coenzyme A. It combines with carnitine at the outer mitochondrial membrane. Then it forms acyl carnitine with the help of CPT1. This acyl carnitine is transported on the matrix side of intermitochondrial membrane with the help of translocase. This is again converted back to acyl coenzyme A and carnitine by CPT2. Carnitine is again transported back with the help of translocase. That's it. So basically, even if all the enzymes involved in the beta oxidation of fatty acid are present in the body, but if there is somehow some problem or defect or deficiency of the carnitine, still the fatty acid beta oxidation cannot happen, because fatty acid is present in cytosol. It has to get transported into the mitochondrial matrix for the reaction to happen. Now we will see, we saw that how fatty acid got activated. After activation, it came from cytosol to the mitochondrial matrix. So now the activated fatty acid has reached mitochondrial matrix. How the beta oxidation will happen? So in the first step, dehydrogenation is happening. Okay, so what is happening basically? Two hydrogen atom is, this dehydrogenation is a type of oxidation. Remember that. In enzyme you would have read, okay, different classes of enzyme. So in that they would have told you. So these two hydrogen atoms are removed from the alpha and beta here. Alright, it will form double bond ultimately. So acyl coenzyme, which is activated fatty acid, it getting oxidized by removal of two hydrogen atoms from alpha and beta carbon, forming trans-enoyl coenzyme. This is, even if you don't remember this, it's okay. Trans-enoyl coenzyme. From acyl coenzyme, trans-enoyl coenzyme is forming, which is known as oxidation. The enzyme involved is acyl coenzyme dehydrogenase. Acyl coenzyme dehydrogenase. Okay. The cofactor required is Fa2, FAD, forming FADH2. Okay. So acyl coenzyme, first proper reaction of the beta oxidation is acyl coenzyme getting oxidized into trans-enoyl coenzyme A with the help of enzyme acyl coenzyme dehydrogenase. The cofactor required is Fa2, FAD, which is forming FADH2. So if in exam it comes, you have to write all the four steps. First you have to write the activation, okay, then you have to write transport, then you have to write four steps. So in here now, whenever the cycle or any reaction or pathway comes, no need to write in words, you can just write reaction. So here of course you don't have to draw ever, okay, you just would write acyl coenzyme, arrow you give FADH2, FAD2, FADH2. Here you can write the enzyme name, okay, acyl coenzyme dehydrogenase, then trans-enoyl coenzyme. This is the first reaction. Second reaction will be hydration. So this trans-enoyl coenzyme water molecule will add and form hydroxyacyl. These things you leave. For you it's okay if you don't write anything about this. So trans-enoyl coenzyme forms hydroxyacyl coenzyme. Simple, hydration is happening, so hydroxyacyl. The enzyme required will be hydratase. Hydration is happening. So enoyl coenzyme hydratase. Enoyl coenzyme hydratase. Water molecule will be required. So trans-enoyl coenzyme one water molecule gets added with the help of hydratase enzyme forming hydroxyacyl coenzyme A. Next step, this is oxidation again. Okay, third step is also oxidation. So hydroxyacyl coenzyme A gets oxidized with the help of NAD plus. There it was FAD forming NADH, and ultimately leads to formation of ketoacyl coenzyme A. Okay, hydroxyacyl with the help of enzyme hydroxyacyl dehydrogenase. So oxidation is happening with the help of dehydrogenase enzyme in the first and third reaction we saw. So in first the cofactor was FAD. In this third step, cofactor is NAD plus, forming ketoacyl coenzyme A. We will see the full reaction together also. This is the last step. This ketoacyl coenzyme A, thiolysis will happen with the help of thiolase enzyme. So ketoacyl coenzyme A, thiolase coenzyme A, SH will be required here, forming acetyl coenzyme A. So two carbon will be released. Two carbon molecule will be released, forming shortened acyl coenzyme, right? Because this is still acyl coenzyme. This is still the chain is still of fatty acid combined with coenzyme ASH, but the two carbon has released in the form of acyl coenzyme. So this is now this shortened acyl coenzyme. So full reaction we will see here. You can see this properly. Acyl coenzyme, here we are not showing how it gets activated. Before that, when in exam it comes, first you have to write here fatty acid, okay, combined with coenzyme A, okay, requiring what? ATP forming acyl coenzyme A plus AMP, correct. Then you will write here transport across the mitochondrial membrane with the help of carnitine, okay, like that. Then you will write in the mitochondrial, this whole reaction is happening in the mitochondrial matrix. In mitochondrial matrix, acyl coenzyme A in first reaction with the help of enzyme acyl coenzyme A dehydrogenase and cofactor FAD forms trans-enoyl coenzyme A and FADH2. The substrate is acyl coenzyme A, enzyme is acyl coenzyme A dehydrogenase, cofactor is FAD, the product is trans-enoyl coenzyme A and byproduct is FADH2, and this whole reaction is known as oxidation. This is a type of oxidation. In next step, this trans-enoyl coenzyme A will incorporate one water molecule with the help of enoyl coenzyme hydratase forming hydroxyacyl coenzyme A. Okay. Now this hydroxyacyl coenzyme A with the help of enzyme hydroxyacyl coenzyme A dehydrogenase requiring NAD plus forms ketoacyl coenzyme A, and for at the same time it forms NADH. This ketoacyl coenzyme A takes up one coenzyme ASH molecule, and with the help of enzyme thiolase forms one molecule of acetyl coenzyme A and two carbon shortened acyl coenzyme A. And now this acyl coenzyme A again will undergo these four reactions, 1, 2, 3, 4. The whole fatty acid chain has been broken down into acetyl coenzyme, right? Acyl coenzyme formed in the cleavage reaction re-enters the, so it is the cleavage reaction. Here it is separating, acetyl coenzyme molecule is separating. So this acyl coenzyme A from the cleavage reaction re-enters the oxidative pathway as reaction 2. So this is actually the reaction 2 because the reaction 1 is the activation of fatty acid, right? So it will re-enter here. So this is what is written here, okay? Since acetyl coenzyme can be oxidized to CO2, so now what will happen to this acetyl coenzyme? This acetyl, how this is going to give energy, now you will ask, right? So this acetyl coenzyme A will be oxidized to CO2 and water with the help of citric acid cycle. Citric acid cycle you would have read, right, in carbohydrate metabolism also. So this acetyl coenzyme A will get utilized in citric acid cycle for the complete oxidation of fatty acid. Now, so this is, so there it will release energy. So acetyl coenzyme A is one source of energy from the beta oxidation of fatty acid. What are the other sources? FADH2. We know that FADH2 is equivalent to 1.5 ATP, right? In electron transport chain, it gives 1.5 ATP, and NADH is equivalent to 2.5 ATP. Okay. So this also will give energy, this also will give energy, and this also will give energy. So this is the energy yield, right? So let's take the example of palmitic acid. So first palmitic acid will get, I will repeat everything. Palmitic acid will get activated with the help of ATP and coenzyme A forming palmitoyl coenzyme A. So here two molecule ultimately two high energy phosphate bonds are released. So 2 molecule of ATP equivalent to 2 molecule of ATP is getting utilized plus 7 cycles because this is 16 carbon, right? So ultimately 7 cycles of the 4 reactions, right? Oxidation, hydration, oxidation and thiolase. These 4 reaction will happen 7 times. So 7 FAD will be required, then 7 NAD will be required. This will be in the second step, this will be in the fourth step, and 7 coenzyme A will be required, right? Coenzyme ASH in the thiolysis reaction, last reaction, plus 7 molecule of water will be required, forming 8 molecule of acetyl coenzyme, 8 into 2, 8, 16. So palmitic acid contains 16 carbon. 8 acetyl coenzyme will form, 7 FADH2 will form, 7 NADH2 will form, and 7 hydrogen. So what will the energy yield? So each cycle, how many are FAD and NHD is releasing each cycle? 1 right? So FADH2 plus NADH 1 molecule will release ultimately which is equivalent to 4 ATP. This is 2.5, this is 1.5. So equivalent to 4 ATP. So in 7 cycle total ATP yield from FADH2 and NADH will be 28. Okay. And now this 8 molecule of acetyl coenzyme will give 80 ATP with the help of citric acid cycle. One acetyl coenzyme releases 10 ATP in the citric acid cycle. So 8 molecule will release 80 ATP in the citric acid cycle. So total will be how much? 80 plus 28 is equivalent to 108 ATP. But 2 ATP gets utilized in the activation. So ultimately the net gain of energy is equivalent to 106 ATP. Okay, so now some disorders we will see. So first is deficiency of carnitine. I told you carnitine is important, or carnitine transferase or translocase. So carnitine is required because carnitine combines with acyl coenzyme forming acyl carnitine, right? Or carnitine transferase because this reaction of combination of acyl coenzyme with carnitine is catalyzed by carnitine transferase, either CPT1 or CPT2, or translocase because this is required for the transport from outer mitochondrial membrane to the mitochondrial matrix. If there is deficiency of either carnitine or carnitine transferase or translocase, this will lead to organic aciduria. Okay, so this generally will manifest in childhood only, in newborn and preterm infant, especially because of inadequate biosynthesis. Okay, if the baby is preterm, it has born before the term of like completion of the pregnancy, the inadequate biosynthesis of these molecules can be there, or it can be because of renal leakage, some problem with the kidney, or in hemodialysis patients also, it will get removed, okay, carnitine. So the patient will manifest with organic aciduria. Symptoms will include muscle cramp, severe weakness, and ultimately it can lead to death also because of hypoglycemia and hypoketosis, because body sometimes needs the fatty acid beta oxidation, right? So if that is not happening for the energy requirement, if that is not happening, then hypoglycemia and hypoketosis can happen. So what to do? What is the treatment? There is no specific treatment as such. The treatment will be basically either to replace carnitine or carnitine transferase or translocase depending on the deficiency. But supportive treatment is diet containing medium chain fatty acid because they do not require carnitine shuttle. Okay, if medium chain fatty acid, long chain fatty acid generally required, but if medium chain fatty acid does not require carnitine shuttle to enter mitochondria. Now, next disease is Jamaican sickness. So, Jamaican vomiting sickness is caused by eating unripe fruit, like from ackee tree, and there are other trees also which releases this toxin. The toxin name is hypoglycemia, especially if you eat a few of the fruits when you are like in empty stomach. So it can lead to hypoglycemia in the body. So it contains toxin hypoglycemia. What it does basically, it inactivates medium and short chain coenzyme A dehydrogenase. So dehydrogenase enzyme is required for beta oxidation. So this hypoglycemia toxin found in few fruits can lead to inactivation of dehydrogenase enzyme which will inhibit beta oxidation and thus leading to hypoglycemia. That's why fruits mainly, because if you don't know whether this contains hypoglycemia toxin or not, fruits should be generally not in the empty stomach. If you are eating after food, then even if beta oxidation is inhibited for the time being, it won't be a problem. And next disease name no need to know much is dicarboxylic aciduria. Only thing is like of interest is this deficiency of carnitine is very common, and Jamaican vomiting sickness. Now what we saw was even chain, right? Palmitic acid was even chain, 16 carbon containing, right? So how what happens in the beta oxidation of odd chain fatty acid? Here all the reactions are same, but at the end it will not form acetyl coenzyme A and acetyl coenzyme A in the last cycle, last reaction, right? Because it is odd chain, so one molecule of 3 carbon compound will form, right? So fatty acid with an odd chain of carbon dioxide oxidized by the pathway of beta oxidation, producing acetyl coenzyme A until a 3 carbon residue remains. At the last 3 carbon will be left because it is odd chain. So that will be propanyl coenzyme A. So what happens to this propanyl coenzyme A? Acetyl coenzyme will go into the citric acid cycle releasing ATP, those things will happen, right? But what happens to propanyl coenzyme? This propanyl coenzyme is converted to succinyl coenzyme, and if you remember citric acid cycle, you will see that this succinyl coenzyme is one of the intermediate of citric acid cycle, right? So how this propanyl coenzyme is getting converted to succinyl coenzyme? Propanyl coenzyme with the help of ATP first will convert to D-methylmalonyl coenzyme, forming L-methylmalonyl coenzyme and forming succinyl coenzyme. That's it. And this succinyl coenzyme will be utilized in the citric acid cycle. And this succinyl coenzyme can ultimately form oxaloacetate and pyruvate, and it can form glucose. So it is basically it is glucogenic. Ultimately, it can act as a substrate for gluconeogenesis. I think you would have read gluconeogenesis in carbohydrate metabolism, right? You know that acetyl coenzyme is not a glucogenic molecule. It cannot form pyruvate or oxaloacetate. So this cannot be utilized for gluconeogenesis. But this succinyl coenzyme is a glucogenic molecule. So ultimately what happens, if succinyl coenzyme can be utilized for glucose formation, we can say propanyl coenzyme is a glucogenic. So propanyl residue of from the odd chain fatty acid, only part of fatty acid, that is glucogenic. Okay. Acetyl coenzyme cannot be converted into pyruvate or oxalate. To go into the gluconeogenesis pathway, any molecule generally has to convert to either oxaloacetate or pyruvate. Then only it can go back into the gluconeogenesis path. So acetyl coenzyme is not a glucogenic molecule, but propanyl coenzyme is a gluconeogenesis molecule. So that was odd chain. What is happens in the beta oxidation of unsaturated fatty acid? No need to know anything more. The reactions are normally same, but two additional enzymes are required in this: isomerase and reductase. Okay, just know that is the difference. Two more enzymes, isomerase and reductase, are required for beta oxidation of unsaturated fatty acid. Energy yield is less because it does not get fully reduced unsaturated fatty acid. So regulation of beta oxidation. So depending on the availability, right? If the more and more free fatty acids are available, more and more free fatty acids will enter the cytosol, that will get activated, and ultimately it will reach mitochondria and more beta oxidation will happen, right? Glucagon insulin ratio. If you remember my last class, I had told that insulin promotes fatty acid synthesis. So it means what? It wants to form more and more complex molecule. In general, if you know, the insulin's action is that it will try to remove simple molecule from the circulation and it will form complex biomolecules inside the cell and keep it, store it. So insulin basically will decrease beta oxidation because it does not like breaking down. Insulin is an anabolic hormone, if you have read, if you had heard, not catabolic. It means what? It does not degrade normally. So it mainly forms, it helps in the forming of bigger molecule. So insulin will inhibit beta oxidation. Because beta oxidation is basically the breaking down of fatty acid. But glucagon is a catabolic hormone. It promotes beta oxidation. So glucagon is to insulin ratio will determine whether the beta oxidation will increase or decrease. If this ratio is more beta oxidation will happen. Now CPT1, I told you, carnitine, palmitoyl, acyl transferase. One is the regulator of entry of fatty acid into mitochondria. So the rate of fatty acid beta oxidation will be mainly determined by CPT1. So if this CPT1 is activated, more and more fatty acid will be transported from the cytosol to the mitochondrial matrix. Now malonyl coenzyme A inhibits CPT1 activity. So from where this malonyl coenzyme A is coming? Malonyl coenzyme A forms from the acetyl coenzyme A in the fatty acid biosynthesis. So if acetyl coenzyme A is forming malonyl coenzyme A, it means that the pathway of fatty acid biosynthesis is required, not the breaking down. So that's why this malonyl coenzyme A inhibits CPT1. So if inhibit CPT1, then fatty acid oxidation will decrease and this malonyl coenzyme will increase fatty acid synthesis. And if this is related to this, if the fatty acid synthesis is going, active fatty acid synthesis is going on, beta oxidation will be inhibited. Some one or two more important points about minor pathways. Alpha oxidation occurs at C2 instead of C3. I told you from the COH side, COH, C of the COH is the carbon number 1. C2, the next carbon to COH is alpha carbon, next carbon to C2 is beta carbon. So like this fatty acid oxidation starts at between C2 and C3, it will be beta oxidation. But if it happens at COH and C2 level, then it is alpha oxidation. So one carbon is removed, right? One carbon atom is removed one at a time from the carboxyl COH end. The process is important in brain. So alpha oxidation, remember in brain it's important. Omega oxidation occurs from the methylene, from the omega carbon. If you remember the lipid chemistry, I told that omega numbering starts from the methylene, right? Omega 3, omega 6, that's how we sell. But if it is delta, then delta we have to number from the COH side. This thing you have to go back and read in book. You will remember. So here in omega oxidation, oxidation starts from the methyl end of the fatty acid molecule. And this happens in endoplasmic reticulum, not in the mitochondria. Remember, endoplasmic reticulum of liver and kidney. Here the enzyme required is hydroxylase enzyme and cytochrome P450 and NADPH, not NADP, NADPH. This pathway omega oxidation becomes significant when beta oxidation is defective in the body. Next is paroxysmal fatty acid oxidation. This occurs for the chain shortening of very long fatty acid chain. Okay, so if any fatty acid chain has very long chain, this paroxysmal fatty acid oxidation will happen. Here the product will be acetyl coenzyme. Ultimately, it will form H2O2 also. H2O2 is not formed in either alpha or beta oxidation. This is induced. Paroxysmal fatty acid oxidation is induced by high fat diet. Okay, and this proximal fatty acid oxidation is also involved in the shortening of the side chain of cholesterol. Okay, cholesterol we had seen, right? It's a big molecule, right? So for the shortening of the side chain of the cholesterol, proximal fatty acid oxidation happen because cholesterol required for the formation of bile acid. So this we saw last time also. You'll see today. What is the difference? Major difference between oxidation and biosynthesis? Site is mitochondria for oxidation, cytoplasm, cytosol for biosynthesis. Intermediate coenzyme A derivatives. We saw na hydroxyacyl coenzyme A, enoyl coenzyme A, okay, ketoacyl coenzyme A, and here all the molecule is the whole molecule till it forms total as fatty acid. It is linked to ACP, right? Acyl carrier protein. Sequential unit, 2 carbon unit acetyl coenzyme is released in each cycle, right? And here 2 carbon unit coming from the malonyl coenzyme. Enzyme, all the enzymes are independent. Here multi-enzyme, coenzyme important here FAD and NAD plus, and here NADPH. And we had seen the sources of NADPH also. So that's it for today. Next class, most probably I will be taking ketone bodies. Okay.
Thank you, sir.
Okay, I'll end the presentation.
Thank you, sir.
Okay.
Thank you so much, sir.
Thank you, sir.
Okay, bye.